This video gives a brief introduction to diffusion MRI and talks through an implementation of diffusion tensor imaging using MATLAB. Diffusion MRI gives a unique probe into the cellular architecture of tissue. It works by sensitizing the intensity of MR images to the dispersion of water molecules through Brownian motion, typically over a few tens of milliseconds. And over a few tens of milliseconds, a water molecule on average will move a few tens of micrometers. And that's the length scale of biological cells. So what determines the dispersion pattern of water molecules over that time is the local tissue cellular architecture. If you think of just a glass of water at room temperature or at body temperature, although the water in there looks perfectly still, actually, if you were to look at individual water molecules, they're all moving around quite rapidly. You can see that if you were to put a drop of ink in the center of that glass, and you'd observe that gradually over time, that drop of ink spreads out over the extent of the glass, as in a way shown down here. And even if you were to just look at the group of water molecules initially at the center of the glass, those water molecules would gradually spread out over the extent of the glass, their initial positions being taken up by other water molecules. And that's precisely the effect that we use in diffusion MRI to probe tissue architecture. To get a clearer picture of how that works, let's think about the human brain and water diffusion in the tissue of the brain. Broadly, three types of tissue in the brain. There are some areas like the ventricles in this kind of T shape at the center here, which contain just fluid without any cellular architecture. So actually there's a substance called cerebrospinal fluid in the ventricles, but for our purposes, it's pretty much equivalent to just water. And the water molecules in there behave very much like water molecules just in a glass of water. So they're free to move around in any direction, unimpeded by cells and other barriers that we see in denser tissue. So the dispersion pattern of water that we see in those areas is, first of all, isotropic, like these pink contours that I'm showing here. So isotropic means looks the same in all directions. A water molecule unimpeded by tissue is equally likely to move in any direction. And also, water molecules will move further on average in that kind of environment than they do in denser tissue, where their motion is impeded by cell walls and other microscopic entities. The second class of tissue is white matter. So white matter is the cabling that allows the brain to talk to itself and to other areas of the body. It even looks like cabling. This is a cross section through a piece of white matter. So it consists of bundles of nerve fibers or axons. These things with the thick black outlines are nerve fibers. And if you imagine a water molecule wobbling around in this kind of environment, it's not difficult to see that it's relatively easy for molecules to move in the directions of the fibers because there's little impeding their motion in that direction, but it's much harder for them to move in the perpendicular directions where they have to, they get impeded by these cell walls, they can get trapped inside the fibers, and even if they're outside, it's difficult for them to move perpendicularly because they have to go in and around all the different cells and fibers. So the kind of dispersion pattern that we see then in these environments tends to be anisotropic. So it's elongated in the direction of the fibers. This particular pathway here in the white matter of the brain is a thing called the corticospinal tract. So it's a big bundle of fibers that runs up the spinal cord into the brainstem and then up towards the motor areas at the top of the brain that control the movement of limbs and other bodily actions. And so the fibers in this, the individual nerve fibers in this bundle then point from the top to the bottom of the brain. And so the dispersion patterns that we observe are then elongated in that same orientation. And vice versa, if we observe an anisotropic dispersion pattern like this, we can make an inference that the fiber direction or the dominant fiber direction is in the orientation in which the diffusion or dispersion is greatest. The final class of tissue is grey matter. So this has a much less coherent structure than white matter, at least at the scale of MRI voxels. Tends to consist of a kind of tangle of cell bodies and processes with no sort of coherent organization. So the dispersion pattern that we see then tends to look isotropic, a bit like the free fluid case that we looked at first, but less widespread because the water molecules still have to navigate around lots of cellular structures, but they're equally impeded in all different directions. So the overall dispersion pattern ends up again being isotropic, but less widespread. Let's just think a bit about how we go about sensitizing the MR signal to diffusion. It's achieved through a particular pulse sequence. This is a pulse sequence diagram in the bottom left here. It's essentially a program that tells the MR scanner how to manipulate its magnetic field during the course of a measurement. And the particular feature that makes this pulse sequence sensitize the MR signal to diffusion are these two blocks here, during which the scanner introduces a gradient into its internal magnetic field. And there are essentially 5 degrees of freedom in this pulse sequence, or 5 device settings using the terminology of earlier lectures. One of them is the length little delta of these pulses. Another is their separation, big delta. And the last 3 degrees of freedom are the elements of this gradient vector G, which has both a magnitude and a direction describing the gradient in the magnetic field. For our purposes, for now, we're going to boil down these 5 degrees of freedom into one single degree of freedom, which we're going to call the B value, or the diffusion weighting factor. And it broadly tells us how sensitive the measurement is to the dispersion of water molecules. It's proportional to the area underneath this gradient. So the bigger the gradient and the longer we leave it on for, the greater the sensitivity to diffusion. And that allows us to write down a first very simple model relating the diffusion MR signal, SB here, to an interesting property of the tissue. And that property will just be its diffusivity, or the diffusivity of water within that tissue. And we're going to call that D. So it relates to the signal through this simple model here. And just a very quick reminder about what diffusivity means. So for a diffusivity of D, it tells us how far on average molecules will move over per unit time. So this is the classical solution to Einstein's diffusion equation that tells us that for a diffusivity of D, the average displacement of a molecule over time t equals this simple expression here. N is the dimension, which is 3 in our case. We're thinking about water molecules dispersing in 3 dimensions. So here's our simple model then. I'm going to write it down on this on the virtual whiteboard just because we'll use it again in a minute. So we are saying then that the signal, the MR signal for a B value of B is equal to the non-diffusion weighted signal, where the B value is set to 0, S of 0, times by this exponential of minus the B value or diffusion weighting factor times the diffusivity. And this tells us that the diffusion weighted signal SB is an attenuated signal of the unweighted signal. This signal S0 is always positive. The B value is greater than or equal to 0. The diffusivity by definition is always greater than or equal to 0. So that means that the term in the exponential minus BD must be less than or equal to 0, which tells us that the exponential itself must be between 0 and 1. So it's in that interval, which then tells us that SB must be in the interval 0 up to S0. So the measured signal for a non-zero or positive B value is always less than the unweighted signal, and it's attenuated by an amount that depends on both the diffusivity. The greater the diffusivity, the greater the attenuation, i.e. the lower SB, and also depends on B itself. The greater B, the lower the diffusion weighted signal. So this immediately tells us, gives us a simple strategy for estimating the diffusivity D. We can rearrange this equation quite simply to write D in terms of things that we know or can measure. Let me first of all just write it again now using measurements rather than signals. So I can write the measurement A for a particular B value B is then the unweighted measurement times E to the minus BD. So we'll use this to estimate D. If we take logs of both sides, log of A, B equals log of A0 minus BD, and we can rearrange to give us a formula that provides an estimate of D from two measurements, one with a B value of 0 and one with a B value greater than 0. Okay, so there's a simple formula to estimate D from two measurements. And we can use that then to make a map of D over an image. Let's switch to MATLAB for a moment to see how that's done. So I've taken the liberty of loading a diffusion MRI dataset into MATLAB here that we can use to illustrate some of these concepts. So before we get started on estimating D, let's just have a look at this dataset that I've loaded in. It comes from the Human Connectome project. You can register with their website and download their data for free. It's just one example from their archive. So the array that contains the actual data is this one here, DWIS. That stands for diffusion weighted images. And let me just show you some stuff about that to begin with. So DWIS then is a 4-dimensional array. It consists of 108 separate scalar images. Each of those images is volumetric, and so it has 3 dimensions. The last index here is the slices, so that it consists of 145 horizontal slices through the brain, and each of those slices is a 2-dimensional image with 145 times 174 pixels or voxels. Let's have a look at so that then this array B-vals contains the B value that was used for each one of these 108 images. Let's just have a look at it. So there it is. And so you can see that some of the images are acquired with a B value of 0, whereas most of them are acquired with a B value very close to 1000. So every 6 or 7 images has the B value set to 0, but most of them have the B value set to around about 1000. Let's just look at the first 2 of these to begin with. So the first one has a B value of 0, and the second one has a B value of 1000. So we can use these first 2 images to execute our initial strategy here of estimating D simply by taking the difference of the logs between these 2 images. And that should produce an estimate of D in each image voxel. So let's do exactly that. Actually, before I do it, let's just have a look at the images themselves. So let me start just by showing you one slice of the first image in that array. So I'm going to manipulate it just to get it in the right orientation. Flip up down is doing a vertical flip of the image. And I also need to squeeze just to avoid singleton dimensions in the array. So I'm going to take the first image in this DWI's dataset then. Every pixel in a particular slice, I'll just take a slice that's somewhere near the middle, number 72. Then I also need to transpose it to get it in the standard orientation, which has the front of the brain at the top of the image. And then the second argument to him show, if you just put open brackets like that, it scales the intensity to the full range of values contained in the image array. So let me just open a new figure for this. Okay, so there is one slice through the first image. This is one with the b-value set to 0. I'll put it here for the moment. And now let's have a look at the corresponding slice in the second image in the data set. This is one with the b-value set to 1000. So there it is, and you can see that the appearance is quite different. In particular, this bit here, this bright bit in the center in the B0 image is the ventricles, where there's a lot of signal when there's no diffusion weighting, but it disappears very rapidly when there is diffusion weighting, because of course the water is free to move there, so we get relatively high diffusion compared to these denser tissue areas outside. Although this image looks brighter than this one, that's only because we've scaled them both to each's intensity range, whereas actually the intensities in this image are much higher than this one, because this one has no diffusion weighting, so there's no attenuation, whereas this is a strongly attenuated version of this image. We'll see that again a bit later. Okay, so let's try then and use these two images to estimate a map of D over this same slice. So we can write then D, let's call it D map. We'll call it D map 72 for slice 72. So simple formula then. So if we just take log of this, if this slice of the first image, so this is the A0 here, B value set to 0. We'll take the log of that slice minus log of the slice with the B value at 1000. That's this one here. And divide by the B value of the non-zero B value, so that would be the second entry in our B vals vector here. That corresponds to the B value of the second image. Okay, so just because performing the operation this way will leave a singleton dimension in this DMAP. I'll show you that just to illustrate. So you see it gets this single dimension at the start, so I'm going to squeeze it in order to get rid of that singleton dimension, which I need to do in order to be for ImShow to be able to display it. So let's start by squeezing it. Now if I do that size operation again, you should see that it's just got 2 dimensions now. There we go. And now I'm going to display it using pretty much the same operation that I did before. So I can get rid of the squeeze already. But I'll do the same flipping and transposing to get it in the right orientation. Oops, one too many brackets somewhere. we go. So there is a map of D. So actually, if we just flip back to the PowerPoint for a second, you'll see that on the next slide, I've done this operation already. And actually, so the map of D that we've got here doesn't quite look like the one that we've just generated. And I'll show you why that is and fix it in a second. The reason is that if we look at the values in this image in the background, you'll see that we're getting not a numbers in the background. And the reason for that is that in these background areas of the image outside the brain, the intensity values, there's very little signal coming from the scanner, and the intensity values are all set to 0. And what that means is, of course, when we take logs of it, log of 0 is -infinity, and so we get not a number in MATLAB. So I'm just going to remove those not a numbers so that we can see the image a bit better. So I'm going to search for all the NANs within this. So we DMAP 72, didn't we? This NAN. So we'll find all of those pixels and set them to 0. So now let's replace that image with this one with those NANs removed. Should look a little better. Still not going to be perfect. So closer, but now we're seeing something else slightly strange, which is that the background, although these should all be zeros, have a sort of mid-grey intensity, and you can see a few sort of black spots appearing. And the reason for that is that in some of these kind of noisy pixels around the edge, we can have signals, the diffusion weighted signals that are higher than the unweighted signals just through random noise, and that can cause a negative estimate of the diffusivity, which of course is physically impossible. So I'm also going to now remove all of the values of the diffusivity that are less than 0. So one more step. That should do it. And now if we display it again, we should get something that looks more like what I had in that slide. So now we get a nice map of the diffusivity where it's 0 in the background, high in the ventricles, and sort of lower in the denser bits of tissue. Right, let's go back to the PowerPoint now. And so there was one simple estimate then of the diffusivity map. One thing you'll notice about it is that it ends up having quite a noisy appearance. And you'll also recall that rather than just having 2 measurements in it, which is really all we need to make an estimate of the diffusivity, the data set I'm looking at has a much larger number, 108 of different measurements. And that's typical in diffusion MRI data sets, even if we're only trying to estimate the diffusivity, basically to give us a better noise control and to remove this kind of noisy effect from the estimate of D. So let's have a better look at the whole data set now. Here is a picture of it. So this is just showing one slice, corresponding slice from each of the 108 images in that data set. And what you can see is that there are several bright ones but a larger number of these darker ones. These are the first 2 here, these 2 that we already looked at over here. This is the first B0 image, and this is the first B1000 image. So now in this image, the scaling is the same for all of these images. So we can see now that this diffusion weighted image is much darker. It has lower intensities than the unweighted B0 image. And in fact, all of these darker ones are diffusion weighted images. They all actually have a B value of 1000 here, and all of the brighter ones are unweighted images with a B value of 0. So there are some labels just showing that. And if we go back to the MATLAB just for a moment, and so you'll recall that the full set of B values looked exactly like that. Almost all of them were set at 1,000, but then every 6 or 7 measurements or so, we have one that's acquired with a B value of 0. So the question then becomes, so how can we use this bigger set of images to make a better estimate of our diffusivity parameter? So one thing that we might do is simply to take take 2 averages. We could take one average of all of these B0 images and use that as our A0. Let me just move that down a bit so you can see it. Use that as our A0 here, and then take a second average of all of these diffusion weighted B is 1,000 images and use that as our AB here. And that is not an unreasonable strategy. It will produce a much nicer diffusivity map than this one, but it's not quite satisfactory because it ignores the fact that there are actually many more of these diffusion weighted images than there are of the non-diffusion weighted images. So actually there are only 18 of these B0 images, whereas there are 90 of the B1000 images. And really we'd like to find a strategy for estimating D that will accommodate that difference in the number of B1000 images and B0 images in order to provide us with the best possible estimate of D. Just to think a little bit further about all of these diffusion weighted images. So in fact, the 90 B1000 images are not all identical to one another. I mentioned earlier the orientation of the gradient vector, and I'm going to call that orientation Q hat. So Q hat is a unit vector in the direction of the gradient. And we can pick that Q hat to be any direction that we like. And typically what's done in diffusion imaging is that we acquire a large number of diffusion weighted images with the B value set equal in all of them, but each of them has a unique Q hat. And that's exactly the case in this data set that we're looking at here. This slightly strange picture at the top here is showing placed on the sphere a little X that marks the direction of each of the Q hat in each of these diffusion weighted images. So each has a unique one. Each one of these X's corresponds to the gradient direction in one of these diffusion weighted images. And they're chosen as a set so that they they sample the surface of the sphere, i.e. the set of all possible directions, as evenly as possible. And actually, if we just flip back to the previous slide, you can see some subtle differences between the diffusion weighted images. So for example, if we look at this pair here, you can see, if you look closely, that in this one here, this little sort of right pointing arrow in the center here that I'm highlighting with the mouse is a structure called the corpus callosum, a white matter structure called the corpus callosum. In fact, if we go back a couple of slides, you can see it on this picture here. This is the corpus callosum here, this from looking at it from a different angle. But it's a big white matter structure that links the left and right hemispheres of the brain. So it's a large structure, and the primary one that allows the left and right sides of the brain to talk to each other. So it consists of fibers pointing in the left-right direction, which is how the connection is made. And so, so we'd expect there to be a strongly anisotropic diffusion dispersion pattern within that region, elongated in the direction of the fibers, i.e. the left-right direction. Left-right on this picture is actually sort of up-down. So this is the left side of the brain, this is the right side of the brain. And so what we see in this particular diffusion weighted image is that we get quite, it's quite bright, that region corresponding to the corpus callosum, suggesting that there's relatively little attenuation of the signal there, suggesting that the diffusivity in the corpus callosum in the direction corresponding to this image is quite low. And that suggests that that gradient orientation is perpendicular to the fibers here. So the fibers are running left, right, so the gradient orientation must be in some perpendicular direction to that. Whereas if we look at the neighboring image here, we can see that the same area has a relatively low signal, suggesting that there's more stronger attenuation of the unweighted signal. So there must be relatively high dispersion in the direction of the gradient, suggesting that here the gradient is more aligned with the fiber direction, i.e. it's a direction more left-right in this direction here. So we see larger attenuation here because the gradient is in the direction of the fibers. Here it's perpendicular, so we see low attenuation. So I was talking about how we might go about estimating the diffusivity from this large collection now of not just 2 images, but 108. And the way that we're going to do that, that balances nicely the number of images of each type that we've got, is to use standard linear least squares. I'm going to get rid of these images so that we can see the virtual whiteboard a bit better. And then I'll try to explain how that's done. So we can now write each of our measurements as the following. Well, let's call it BK log S0 minus BD. And I'm now going to consider that there are 2 unknowns in this system. There's D, the thing that we're actually interested in, and there's log S0. So I think this is an unknown, although we can make a measurement A0, that's obviously a noisy measurement of the signal S0, and the signal itself remains an unknown that we're going to try and estimate. It's actually kind of a nuisance parameter in this situation. We're not particularly interested in the value of S0, but we need to estimate it in order to estimate the thing that we are interested in, which is D. And so we get a similar linear equation like this by taking the logs for each of our different measurements, and we can write those in a standard system of linear equations as a matrix equation. So we can construct a vector A containing all of the log measurements. So I have log A of B1, log A of B2, all the way down to log A of B108. So there is our list of measurements A. Then we've got a vector X of unknown parameters, and I think we have 2 unknown parameters here. We've got log S0 and we've got the diffusivity D. So now to complete this equation, we just need a design matrix G, which relates our unknown parameters to the measurements that we've made. And it's fairly clear from this equation here that the coefficient of log S0 is simply 1. So the first column of our design matrix G just contains 1s. And then the coefficient of D is minus BK here. So the design matrix then contains minus B1, minus B2, all the way down to minus B108. And if we can, once we construct the design matrix like that, we can then solve this linear system of equations in the usual way in order to obtain estimates of the unknown parameters X. So let me jump to MATLAB again, and we will implement this idea just to illustrate. So let's start by just looking at one individual voxel. I'm going to call it a vox. And we'll pull out one voxel, all 108 measurements corresponding to that voxel. We'll pick one somewhere in the middle. So there is a voxel, set of measurements. Oops. Oh, just a comma. Let's have a look at it. So there is the list of measurements, the 108 measurements from within that voxel. And you'll notice, if you look at this carefully, that every 6 or 7, we have a higher value of sort of 3,500 or so, compared to the intervening measurements, which are typically, 500 up to 2,000, something like that. So you'll immediately realize that these, of course, are the B0 measurements, whereas the smaller ones are the B1000 measurements in between. Okay, so let's then construct this design matrix of ours so that we can, so this is that AVOX is, we need to take logs of it in order to get our A vector here. We'll do that once we've constructed this G matrix, which will then allow us to estimate this vector of unknowns X. Okay, so G then will contain, the first column will be a column of ones of length, the number of B values. Should be minus B values. And then, yeah, the second column is minus the list of actual B values. I need to transpose it to get it into the right orientation, but that then is G. Let's have a look at it again. So there it is, as we'd expect. So just a list of ones in the first column and the B values in the second column. So then very straightforward then to estimate X. Now that we've got G, we simply matrix divide our design matrix G by our measurements, which is going to be, obviously we're taking the log of those measurements, the log of a box. So that's what it gives us. So this is, so these are the values then of log S0 here, the first one, and the diffusivity here, the second one. Just as a sanity check, recall that in the AVOX list, the B0 measurements were all coming out with a value somewhere between 3,000 and 3,500. So I think we should find that if we take the exponential of this, which should give us our estimate of S0, it should be somewhere in that range. So let's just check that. X of X1 is 3,250 odd. Okay, so that's an encouraging sanity check. So that's performed this estimate then for a single voxel. How do we do it for the whole image? Well, we simply now just have to loop over the whole image and perform this same operation for every voxel of the image. So let's do that. What shall I call it this time? I'll distinguish it from the previous one, which we call DMAP72. I'll just call it DMAP72-2. Of course, zeros. So I'm going to create an empty matrix, first of all, to put the answers in as we compute them. So it needs to be the size of the image slice, that's 145 by 172, and then have an extra dimension of 2 elements, because I'm going to put both elements of X into this array as we calculate them. Okay, so then looping over the image, I is 1 to 145, J 1 to 174. So let's get at this particular pixel, we'll get the measurements out of the big DWI's array. We're just focusing on slice 72, of course. And this time, rather than sort of worrying about those background pixels, I'm going to check at each voxel whether there are any measurements that are 0 or less. And if there are, I'm just going to ignore that voxel because there'll be background ones anyway. So I'm just going to skip them. So if the smallest measurement, only if the smallest measurement in A is greater than 0, am I going to process it. Otherwise, I'm just going to ignore it and leave that voxel filled with zeros. So if it passes that test, then I am going to put in this corresponding slot of DMAP72.2, the output of our matrix divide GMAP, G divided by log of A, the parameters that we're trying to estimate. Okay, end the if, end the for, end the other for. Okay, so that's looped over that image slice then and should have solved this matrix equation for every pixel within. So we can have a look at the results. We might still get some negative values of diffusivity, so I think I might just remove those before we look at it. Oops. And set those equal to 0. So wherever it's less than 0, I'm just going to set it equal to 0. Okay, so now let's have a look at this new. Actually, we need the squeezes again, don't we? So let's look at I want to look at the second element, which is the diffusivity. I'm not too interested in the map of S0. So we're just going to look at the second element of DMAP72.2, which should be the diffusivity estimate of the diffusivity here. And there it is. So hopefully you can see that. Actually, I can bring up the old one as well, just for comparison. Where was it? That's the old one. So that was our first estimate of D from just the 2 images. And this is the one that we get from all 108. And I hope you can appreciate that you get a much cleaner and less noisy map from using all 108 images, which you can do very neatly via this linear least squares operation. So one last thing to do, I think just going back to the single voxel that we looked at, having now computed these parameter estimates, I'm going to close these images, we can, of course, plug them back into our signal equation here, this one here, let's see, in order to get estimates of what the actual signals should be that our measurements are noisy versions of, and compare those estimated signals to the actual signals that we measured, partly as a sanity check, but also to get an idea of how well our model is approximating the actual measured data. So very simple to do that. So I'm going to just estimate. I'm going to construct an array A estimates, which would then be, so it's just, I think we just simply have to multiply G and X and take the exponential to get our estimated measurements, because G times X, of course, gives us the list of log measurements. So then we take the exponential to give us estimates of the actual measurements. So let's do that. Let's have a look at A est. There it is. So it's giving us now the exact same value for every one of the B0 images, and similarly the exact same value for every one of the diffusion weighted images, which is of course what our simple model here predicts. And I can even plot those on a figure together with the actual measurements. So let's plot our original measurements, A Vox, on a figure. I'll plot them as, which we do blue squares. So there they are. Oops. Just as a function of position in the array. And let's now add to that figure the estimates that we've got from our model after fitting the parameters to those data points. So I'm going to plot the, oops, no, so that this time I want the AEst, the estimated measurements from the model. I'm going to plot those as red crosses. So there they are. And so as we'd expect, our model thinks that all measurements with the same B value should be the same, whereas actually the measurements that we observe are much more widely scattered, of course, and probably that's because there's some anisotropy, which our simple model here is not accounting for. We can also work out an error score simply by summing, let's call it sum of square differences, summing the square difference between A Vox and A Est. And so that gives us a sum of square differences of, what's this number here, 16 million, I guess. So we'll compare that with later models that should approximate these data better. Not a particularly meaningful number in its own right, but we'll come back to that a bit later. And actually, if we just now flip back to the PowerPoint, I've got some more examples of that. So these are maps on a slightly different slice of a different data set in the PowerPoint, showing the map of S0 and the map of D. And on the next slide, oops, showing various different voxels in the image. So here's one in CSF. We see that first of all, the S0 measurements have a relatively high value compared to white matter and grey matter voxels, so about 4 times the size. And there's a much greater difference between the B0 images and the B1000 images, because we get large attenuation because there's high diffusivity in these regions. But also our simple model that assumes no anisotropy and that all the diffusivities, all the diffusion weighted images are the same at the same B value, actually gives a good approximation to the measurements in CSF, because the dispersion is isotropic in CSF voxels. In grey matter, we see a little bit of dispersion around this sort of constant estimate of the diffusion weighted signal. But in white matter, we see a much higher variation around that signal, and that's kind of reflected in the sum of square differences. Here comes out as 16 million compared to about 4 million, errors of about 4 million in the grey matter and the CSF voxels. So that observation leads us naturally into the next model that I want to discuss, which is the idea of diffusion tensor MRI. Let me just move this figure out of the way. Scroll down and give us some space here. So diffusion tensor MRI is a direct extension of the simple model that we've just looked at for estimating a single scalar diffusivity. It's a direct extension of that to 3D, and now allows the diffusivity to vary as a function of direction. And more specifically, it models D as a quadratic function of direction via this expression here. So D is now a function of Q hat, the orientation, and specifically we get the D for a particular orientation by expanding out this quadratic form. Q is a unit vector, and D is the diffusion tensor, which is this 3x3 symmetric matrix that I've drawn here. And the model then that relates the diffusion tensor to the diffusion weighted signal, which we're now writing as a function of both B, the B value, the diffusion weighting, and Q hat, the gradient orientation, is quite similar really to the previous model that we looked at, but we've replaced the single scalar diffusivity D with this quadratic form that expresses D as a function of direction. So let's have a think about this model then, and how we might go about estimating it. So we can linearize this model in much the same way as we did with the single scalar diffusivity. So first of all, let us take logs of both sides of that equation. And doing that, I can then write log of S B Q hat is log of S0, oops, log of S0 minus B times this quadratic form here that we've written. Let's expand out this quadratic form. So in its writing it out in full, Q, of course, is a 3 vector. And when we transpose it, oops, when we transpose it, it becomes a row vector, and I'm going to call it QX, QY, QZ. So those are the 3 elements of Q. Then the diffusion tensor, of course, looks like this: dxx, dxy, dxz. Symmetric, so another dxy there, dyy, dyz, dxz, dyz, dzz. And then we've got our untransposed Q vector here. So I'm not going to go through every step of multiplying this out, but I'll just write out what happens if you multiply out this matrix and vector expression here. And what we end up with, it's pretty easy to see if you just multiply them stage by stage. We end up with qx squared dxx plus 2 qx qy dxy plus 2 qx qz dxz plus qy squared dyy plus 2 qy qz dyz plus qz squared dzz. So that's what it expands out to. Oops, away. And that shows us then, if we were to plug that back into the original logged equation up here, we can see that now we have an expression for the log signal, log SBQ hat, that relates that linearly to all the elements of the diffusion tensor, dxx, dxy, dxz, and so on. And that shows us then that we can write this as a nice, simple set of linear equations in our unknowns, the elements of the diffusion tensor, to estimate them from our log measurements. So I'm going to write out again that, in a very similar way to we did just a minute ago with the estimating the scalar diffusivity, a matrix equation then that will allow us to estimate these unknown quantities. So again, our A vector, much like before, is going to be just the log signals, log A of B1 Q hat 1, log A B2 Q hat 2, all the way down to log A B108 Q hat 08. So there's our measurement vector. Now let's list our unknowns over here. So our list of unknown parameters is a bit bigger now. First, we still have log S0. That's our first unknown. But then we've got each of the elements of the diffusion tensor. So we have dxx, dxy, dxz, dyy, dyz, and dzz. So we've now got 7 elements of our vector of unknown parameters that we're trying to estimate. And lastly, let's construct this design matrix, which we can do from this expression that we've just generated here. So we can see from up here that the coefficient of log S0 is simply 1. So the first column of our G matrix is, as before, is going to be a big list of 1s. 1, 1, all the way down to the 108th 1. And then so the second unknown is dxx, and we can see that the coefficient of dxx then is going to be, so we've got a qx squared from here, but we've also got a minus b from up here. So I think the full coefficient for dxx then is going to be minus b times qx squared. So I'm going to expand this a bit so I'm going to have to write small to get all these columns in, but it will be, so in the first row, that's for the first b value, I'm going to write that minus b1 q1x squared. And then on the second row, of course, it will be b2 q2x squared all the way down to the last one being minus b108 q108x squared. Then similarly, so the next unknown is dxy. Its coefficient is going to be minus b times 2 qx qy. Let's write that in. Minus b1, oh sorry, should have a 2 there, shouldn't we? q1x, q1y. Next one is dxz. That's going to be similarly minus 2 b1 q1x, q1z. Then we've got the dyy term. That will be minus b1 q1y squared. Then we've got the dyz, that will be minus 2 b1 q1y q1z, and finally the zz term minus b1 q1z squared. And similarly, so and then each row will have the same structure. Let's zoom out again and I can complete this picture. So there's our big design matrix. That's what it looks like. And once we've got that, of course, we can then solve for the unknowns in exactly the same way. So let's have a look at implementing that in MATLAB. So first thing to do is to construct this big G matrix. A bit more to do than last time, but here goes. So the first column then, of course, as before, is a list of ones of length, the number of images, or number of b-values. I'll just use the length of the b-values array. And then the second column then is going to be minus the b-values array times. Now I've got all the q hats in a vector called q hat. Oops, sorry, no, dot times. So dot times does an element by element multiplication. So we're going to multiply the whole array of b values with a whole array of q1 of qx's. So all the q vectors are all in a vector called a matrix called q hat, and the first element of that is the x's. So I can get this column simply by multiplying the list of q hat x coordinates with the list of b values. So that's that. Actually, I won't do the square. I'll just write it out fully like this, just to make it clearer. So that's that second column. Then the third column will be minus 2 times b vals dot times. Now we want qx, which is this one, times qy, which is the second column of q hat. And we're on to the dxz coefficient. Looks like that. And now we're on to the dyyy column. So that's going to be minus b vals. We want q hat 2 twice, don't we? So then the dyz. And finally, the dzz column. I think we're done. To transpose it again, remember to get it in the right orientation. Okay, let's have a look at G. Maybe we'll just look at the first few rows of it, just as a sanity check that I haven't typed something in wrong. Entirely possible. Okay, so it looks sensible enough. Of course, you know, the first column should all be ones, as we expect, and the subsequent columns should all be, you know, something between 0 and 1000. And so they all look like kind of sensible numbers. The first row, of course, corresponds to a B0 image. So all of these, the B value is 0 in all of these, whereas the second and third are all non-zero B values of 1000. So our G matrix then looks sensible enough. Let's start then just with the single voxel that we singled out last time. Remember, we looked at we put all the measurements in an array called a vox. So let's just solve it for that one single voxel to start with. And we can do that in exactly the same way, of course. So we simply matrix divide G by the log measurements. And let's see what comes out. So, okay, we've got the right number of elements, first of all. So remember, we should have 7 numbers here. The first one here is log S0, and it's exactly the same number that we got before for this particular voxel. So that's encouraging. Log S0 comes out the same. And then, of course, these remaining 6 numbers are all the elements of the diffusion tensor. So let's actually construct the diffusion tensor from these numbers just to have a look at it. So the first one, of course, is XX. So we put that in the sort of top left corner. So the top row of the diffusion tensor, let me write it out. Oh, I've got it up here, haven't we? Here it is, up here. So I'm just going to copy all of the numbers into the right positions to get them into a MATLAB array. So DXY is number 3, DXZ is number 4. That's the top row. Then on the second row, so we've got DXY, it's number 3, DYY, that must be number 5, and DYZ, that must be number 6. And then finally, the bottom row starts with DXZ, that was what, number 4. The YZ, that was number 6, and finally DZZ is number 7. So there's our diffusion tensor. There it is. That's what it looks like for that that particular voxel. I can once more, of course, feed that back into our model to estimate the signals back given our fitted parameters. I can do that in exactly the same way as we did it before. In fact, I'll just do exactly that. So est, we just plug it back, multiply the estimated parameters by G to get the estimates of A. And let's actually add them. There's that window gone, the plot. Oh, I still got it, there it is. Let's add them onto this plot to see how they compare with the estimates from our previous model. So that's what we did before. Let's put them as green crosses this time, just so we can distinguish them. There they go. So I hope, let's see if we can make this bigger. So hopefully you can now see, so the green crosses have obliterated the red crosses for the estimates of the B0 signals, because they're exactly the same. But if we look at the diffusion weighted signals with the B values of 1,000, now we can see that unlike the previous model where it just estimates the same number for every single one, now that we've got this diffusivity as a function of direction by the diffusion tensor, you can see that the green crosses, the estimated signals vary quite widely and actually much more closely approximate the blue squares, which were the original measurements. So that's encouraging, and we should also be able to compare the sum of square differences. If you remember, it came out as something like 16 million previously, didn't it? And so now that's dropped substantially to a value of 4 million, showing that this new model with the directionally varying diffusivity fits those measurements much more closely than the previous one. Okay. So next step then is to map the diffusion tensor over a whole slice. So we're going to do that in a very similar way to the way that we mapped just the diffusivity. So we just type that in here. So again, we'll start by creating an empty vessel in which to put the estimates in each pixel. So now, of course, we are going to estimate 7 values in each pixel. So we need an array of this size this time. I'm going to, this time, I'm going to use this tick-tock construct, which allows me to time the execution of a particular series of commands, basically because the first implementation I'm going to show you is inefficient in one particular way, which I'll come back to. And I'll use the timing just to show the extent of that inefficiency. But anyway, we're just going to, as before, loop over every voxel for I is 1, 2, 1, 4, 5. J is 1, 2, 1, 7, 4. Let's pull out the measurement in the corresponding voxel. And we'll do the same test that we did last time to avoid noisy background voxels that have got negative or 0 measurements in them. So we'll only focus on ones where all the measurements are greater than 0. And then we will place the output of our matrix division into the corresponding cells of our DT map. So do the matrix divide in exactly the same way as we've done before. So and then at the end of this last for loop, I'm going to do the tock command, and that will tell us how long this whole operation takes. So here it goes. Okay, so it ran that whole slice, fitted the diffusion tensor in every voxel of that slice in what's that, 651 milliseconds. So let's just keep a record of that just to compare it with another implementation in a second. Actually, I'll tell you what, we'll just do that other implementation now. The inefficiency here lies in this command here, because if you think about it, what is that command actually doing? It's computing the pseudo inverse of G. Looks like this. So G pseudo inverse is, oops, G transpose G inverted times G, and then it multiplies that matrix by the log measurements. But the inefficiency is that, of course, G never changes, and we are performing this, relatively expensive matrix inversion operation every time round this loop. So we're doing it 145 times, 174 times, whereas actually the answer comes out the same every single time. So could we not just compute that a priori and then use it each time around the loop? We can, of course, do that. And so let's do that and see how much difference it makes to the time that it takes to process this whole slice. So I can construct the matrix G_inv, which will be exactly this matrix here. There's a function in MATLAB called pinv, pseudo inverse, that does exactly that. So let's compute this pseudo inverse of G a priori, and then we'll do all of this stuff again. But rather than doing the / operator, we'll write it out more explicitly. It's just G_inv times log A. And we can cut and paste the last few ends of the talk. And you see, so it saves about half the time. It takes about half the time. It's not a dramatic saving, so it doesn't actually take very long anyway. But sometimes that can be important. If you're processing a very large image, sometimes you find that you have to repeat the processing time and time again to estimate uncertainty, for example, which we'll come back to a bit later in this series. But worth doing anyway. So back to our map of the diffusion tensor. So of course, we've now created this object DT map 72. Let's have a look at what's in it. So we can, first of all, we can have a look at the the diffusion tensors that we've created. So we've seen this one for this one particular voxel. We can now map the elements of the diffusion tensor over this whole slice. So let's have a look at that. Let's have a look at what the contents of the, I'll use one of these. Let's use this. So I now have to, I'm going to have to squeeze DT map 72. Let's have a look at, so remember the first element of the DT map array is going to be just the log S0. It contains these 7 numbers in X, of course. So let's have a look at DXX instead, and so that's going to be element number 2 of the DT map array. So this is a map then of DXX. So there it is. And so it looks very much like a diffusivity map, which in fact is exactly what it is. DXX is the diffusivity in the X direction, which is in our frame of reference, left to right in the brain. Some difference in that here we can see, so this sort of slightly, the ventricles are the very bright bit as usual, have high diffusivity there. But there's this other kind of bright bit surrounding it. That again is the structure that I mentioned earlier. It's the corpus callosum. And the reason that that comes out bright in the DXX map is that, of course, these are fibers that point in the left-right direction. And so there'll be high diffusivity in the direction of the fibers, which is exactly the X direction. So I would suggest that if we look, say, at DYY or DZZ instead, let's do exactly that, we should see a difference there because the diffusivity in the corpus callosum in the Y direction, for example, so this would be element 5, 1, 2, 3, 4, 5, this should be DYY. We should see there that there's relatively low diffusivity in the corpus callosum because it's now mapping diffusivity in the front to back direction, which is perpendicular to the fibers. Let's have a look at one of the off-diagonal elements. Let's have a look at DXY, for example, so element number 3. And you'll see that these look a bit different. So for one thing, the background is grey because these off-diagonal elements can be positive or negative. It's only the diagonal elements that have to be positive. And yeah, so they have this kind of, you know, slightly different appearance. But there's no particular, I mean, they loosely reflect the sort of correlation of diffusion in the X and Y directions in this case. But there's no sort of clear interpretation of these images. Let's jump back to our PowerPoint for a second and think a bit more about what this object, the diffusion tensor, tells us. So actually, before we do that, so this is just equivalent to the earlier figure that I showed, showing the diffusion tensor fit to the data in the 3 sample voxels that we showed. So this is, we've already seen this because I generated it, but we get a much lower error score in the white matter voxel and you know, clearer fit of the estimated parameters, the estimated measurements to the actual measurements. In the CSF and the grey matter, there's much less difference, and actually we only get about a 10% reduction in the error scores for both of these voxels, because of course the diffusion is reasonably isotropic in both of these regions anyway. So having the anisotropic model doesn't help particularly. Only really helps in the white matter voxel, where we get this dramatic reduction in the errors from, it was 16 million before down to 4 million here. So here I've then mapped out these all elements of the diffusion tensor in the same sort of structure as the matrix itself. So this is the map of DXX, this is the map of DXY, and so on. And yeah, so we've seen this already, but these are the diffusivities in the X, Y, and Z directions, and these off-diagonal elements, don't really tell us anything that directly. So, the diffusion tensor itself isn't a particularly useful object in its own right. And what we tend to do is to derive various quantities from it that are a little bit more meaningful. So we'll get back to some of those in a minute, but before we do, let's just think a little more about what this diffusion tensor is and what it can tell us both about the dispersion pattern and about the underlying tissue. So there's an inherent implicit assumption in diffusion tensor imaging that the dispersion pattern is a trivariate or three-dimensional Gaussian distribution. And under that assumption, the diffusion tensor then is proportional to the covariance of that three-dimensional Gaussian distribution. Now, a Gaussian distribution in 3 dimensions has ellipsoidal isocontours. And these ellipsoidal isocontours are quite a nice way to visualize and think about the dispersion pattern and the diffusion tensor. So we'll come back to that in a second, but the diffusion tensor itself is a positive definite symmetric 3 by 3 matrix. So symmetric 3 by 3 matrix we know already. Positive definite is obvious if you think about it for a moment. If we flip back here, positive definite means by definition that for any q hat, if we expand the this quadratic form, it must produce a positive value. And that has to be true because it makes no physical sense for the diffusivity to be negative. And so only with a positive definite D can we ensure that every diffusivity comes out positive. So D has to be positive definite. We can derive an eigensystem from D, and so it gives us 3 eigenvalues, and those eigenvalues are the relative lengths of the major and minor axes of these ellipsoidal contours of the dispersion distribution. So the largest eigenvalue then is the relative length of the longest axis of the ellipsoidal contours. The smallest eigenvalue is the length of the shortest axis of the ellipsoidal contours. And the eigenvectors of D define the orientations of those major and minor axes. And lambda 1 then, the primary eigenvalue, is the diffusivity in the direction of the major axis of the ellipsoid, the first eigenvector E1. And of course, in regions where we think there's fibrous tissue, E1 is the direction then of greatest diffusion, and that gives us an estimate of the fiber direction. There's a few sort of standard canonical shapes of diffusion tensor. So if all 3 eigenvalues are approximately equal to one another, then those ellipsoidal contours are approximately spherical. And of course, that gives us an isotropic diffusion tensor. And these are typically what we observe, of course, in CSF, cerebrospinal fluid, and in grey matter. Jumping to the bottom first, so suppose we have 1 large eigenvalue and 2 smaller ones, then that gives us what we call a prolate diffusion tensor or ellipsoid, in which it's elongated in one direction. So we have 1 long axis and 2 short ones, a kind of cigar-shaped ellipsoid. And that, of course, is the typical shape that we observe in white matter when the dispersion pattern is elongated in the fiber direction. But there's, of course, a third sort of canonical shape, which is oblate, and that's when we have 2 large eigenvalues and 1 small one, so that the diffusion tensor or ellipsoidal contours become kind of pancake or frisbee shaped. And I've labeled that white matter as well here. And so how can that arise in white matter? Well, if you think about it, the size of the fiber and cellular structures that we're thinking about are much, much smaller than the size of an MRI voxel. So an MRI voxel would be typically, a couple of millimeters across in each of its sides will be approximately a couple of millimeters typically. Whereas the individual fibers that we're thinking about, these nerve fibers, are more typically something like 1 micrometer across, or maybe a couple of micrometers. So you can have literally millions of these fibers in every single MRI voxel, and they can take a range of weird and wonderful configurations. In fact, it's quite rare that they would all be parallel to one another and pointing in the same direction. When they are all parallel and pointing in the same direction, we get this sort of perfectly prolate diffusion tensor, but other configurations can lead to other shapes of diffusion tensor. And an example that can lead to an oblate diffusion tensor might be when we've got some fibers that are pointing in this direction. So these fibers are all pointing in this red direction here, but then maybe elsewhere in the voxel, we've got a bunch of fibers that are pointing in a completely perpendicular direction. So down the bottom here, they're all pointing in this, this green direction here. And so we'd expect quite high diffusivity in both the green and the red direction, but in the third perpendicular direction, this blue direction here, that's perpendicular to both these, the orientations of both of these groups of fibers. And so we'd expect to have low diffusivity, and overall that would lead to this kind of oblate shaped diffusion tensor. So that's how that can arise. So going back then to the various, I was talking about deriving various more intuitive quantities from the diffusion tensor that we can use in a less cumbersome way than the full diffusion tensor itself. I'll just touch on a couple of these. So the first one is the trace of the diffusion tensor, or the mean diffusivity. So the trace is just the sum of the 3 eigenvalues, or the mean diffusivity is just that divided by 3. So it gives an average diffusivity over all directions. It looks very similar to the single diffusion constant that we estimated with our first model, and in fact is almost equivalent to that. But it's a very useful map because it gives us a sort of overall idea of the mobility of water molecules at each position in the image. It can highlight some interesting pathologies. For example, in diseases that are, attack and kill cells, things like neurodegenerative diseases, they tend to, as cells die, you reduce the number of barriers to the diffusion, so water becomes more mobile and the mean diffusivity will go up. In certain cancers, they can cause a proliferation of very rapid growth of small, dense collections of cells that can impede water mobility more strongly than healthy tissue does, and that can cause the mean diffusivity to go down. So it can highlight different kinds of pathologies. A second common feature derived from the diffusion tensor is, well, there are various measures of anisotropy. Here's just one called the fractional anisotropy. This particular one really is sort of a measure of variance of the eigenvalues of the diffusion tensor, and so gives a measure of how elongated or how far from isotropic each diffusion tensor is. And you can see here that this map picks out areas of white matter. These two V sort of arrow shapes here are once again are the corpus callosum. We get high values of the fractional anisotropy, as we see in various other white matter regions, whereas in the ventricles, the CSF regions, and various bits of grey matter, you see much lower values of the anisotropy because there's more isotropic diffusion. And again, this can highlight interesting pathologies. Typically in diseases that attack the white matter, things like multiple sclerosis, for example, you often see reductions in the fractional anisotropy because where some of these nerve fibers die and then allow more perpendicular diffusion than would be observed in healthy white matter. One more interesting thing we can add to that picture is to weight the fractional anisotropy figure with the orientation from the primary eigenvalue of the diffusion tensor. And so to produce these figures, we simply map the absolute values of the contents of the first eigenvector of the diffusion tensor to the red, green, and blue channels of the color image. So the X component becomes the red channel. So if, for example, you have an eigenvector which is 1, 0, 0, i.e. it points in the left-right direction, the corresponding image pixel becomes very red. And that's what we see in the corpus callosum, because of course it's linking the left and right sides of the brain. If we have one eigenvector that's pointing in the Y direction, that comes out green. Ones that are pointing in the Z direction, this is the corticospinal tract, which you'll recall from earlier, coming up from the brainstem to the top of the brain. And so the primary eigenvalue is in the Z direction, and it comes out blue. Okay, so let's just go back to our MATLAB for a second. We'll just think about computing some of these interesting metrics. So the mean diffusivity is pretty straightforward to compute, of course, because it's just the sum of the three diagonal elements of the diffusion tensor. So I can adapt this command just above really quite easily and sum the three of them. So that this is the second element, of course, was dxx. The fifth one was dyyy. And then the seventh one is dzzz. So if we just add those three maps together, it will produce a map of the trace of the diffusion tensor. There it is. So it looks much like what we saw on the slide. Let's think about the fractional anisotropy. And actually, let me just write down the formula. Let's go back to the PowerPoint just for a second. I have to copy it from there, make sure I don't get it wrong. So the fractional anisotropy, then we can write as, let's get my black pencil back. So fractional anisotropy is, so what have we got? 3/2 times the sum of eigenvalue minus mean eigenvalue squared over sum of eigenvalues squared, all square rooted. So there's our formula. Let's go back to the MATLAB. Let's start by just implementing that on the single diffusion tensor that we already computed. So if you remember, we'd made this diffusion tensor before from the AVOX list that we just picked randomly from the middle of the brain. So there's a function in MATLAB that will compute the eigen system of a matrix. So let's use that. It's called eig. So if we compute the eigen system of that diffusion tensor, it gives us these two matrices, A and B. So B has the three eigenvalues on its diagonal, and A has the corresponding eigenvectors as its columns. So the largest eigenvalue is this one here, and the corresponding eigenvector is this one here. So this would be, for that particular voxel, this would be our estimate of the dominant fiber direction. Okay, so now to try and use these values then to estimate the fractional anisotropy. So I think we can write FA is then, so it's going to be square root of 1.5 times, right, let's do the numerator first. So that's going to be sum of, now there's a function diag in MATLAB, which if you give it a matrix, I'm going to give it B, just pulls out the elements on the diagonal of that matrix. So that will give me the vector containing these three eigenvalues. And then I want to subtract from each of those the mean of diag B again. So that is giving me this term here. Oh, I need to square it, don't I? Yeah. Squared, okay. So that's, yeah, so that's the numerator. And then we need to divide by sum of just diag B this time, isn't it? All squared. Okay, and I need to close. Ooh, I need to close that sum there, and then will I close the square root? Yes, I know. Okay. Okay, so it gives us a fractional anisotropy of. Let's see, that can't be right. Something's gone wrong here. Because it's greater than 1, so let's just read that. Got some brackets in the wrong place. It should have 1 here. That's better than squaring those. So got 1 too many there. I think that's correct now. Yeah, okay. So that's come out as 0.5 now, which is more sensible. Okay, so let's now then compute this, map this over the whole image. And I'm going to do this just by looping over all of them. So we'll create a repository once again. Of zeros, FA, and then we're going to loop minus 1, 2, 1, 4, 5. There's 1, 2, 3, 4. And then we'll pull X from the DT map. And then use the command I wrote out earlier, which turns X into the diffusion tensor structure. Where was it? This one. So we'll construct the diffusion tensor from X, and then we can use these various commands I wrote up above for computing the fractional anisotropy. So we'll compute the eigensystem, and then we'll work the anisotropy out and put it into the corresponding position of the FA array. Just cut and paste that from here. Let that go. Okay. And then let's show it in our usual way. There it is. Nice anisotropy map. So I just want to do one last thing before we end, which is to do a slightly more complex estimation of the diffusion tensor using weighted linear least squares rather than the standard linear least squares, which we've used up to now. Why would we want to use weighted linear least squares? Well, let me try to motivate that to start with. So, of course, using standard least squares makes the assumption that that we've got identically distributed Gaussian noise on all of our individual measurements. So it's actually not an unreasonable assumption that the measurement, each measurement we make, let's call it, sorry, start that again. Oops. So each measurement that we make, let's call it AK, follows a Gaussian distribution around a true corresponding signal, sk, with standard deviation sigma that does not depend on k. So I'm saying then that a Gaussian noise model for the MRI, diffusion-weighted MRI signal, where the standard deviation is the same for every measurement. It's not perfect, but it's not an unreasonable model for that data, which would suggest then that linear least squares, standard linear least squares, is a reasonable way to estimate the diffusion tensor. But of course, we did something slightly unusual, which was before doing the linear least squares, we took the log of all of the measurements. And so the question arises, well, if this assumption is reasonable for the measurements, is it reasonable for the log measurements? So let's have a think about that. So we are saying then that we think that AK is the corresponding signal plus some small perturbation, where delta SK, the noise perturbation, follows a standard a Gaussian distribution with zero mean and standard deviation sigma that doesn't depend on K. Now let's take logs of this. So then log of AK equals log of SK plus delta SK. And let's take a Taylor expansion of that right-hand side. So I can write that then as log of SK plus d by dsk of log SK times delta SK plus some higher order terms. Well, d by dsk of log SK is just 1 over SK, so I can write that then log SK plus delta SK over SK plus higher order terms again. And that tells me then that the perturbation on log AK is delta SK, which of course was Gaussian distributed, divided by the signal SK. And so one might suggest then that the perturbation on, that delta log AK follows a Gaussian distribution with zero mean and standard deviation sigma over SK. But of course, SK is not the same for every measurement. That's the point of using diffusion tensor imaging, is that we think the different AKs vary among themselves. And so that suggests then that the standard deviation in our Gaussian noise model varies for the log measurements, even though it doesn't for the raw measurements. And that's the motivation for using weighted linear least squares, because it allows us then to account for this variation in the standard deviation of the log measurements. So you'll recall from the previous lecture that the difference between standard least squares, where we compute the pseudo inverse G transpose G inverse times G as our mapping from the unknown parameters to the from the measurements to the unknown parameters, so that's standard least squares. In weighted linear least squares, we introduce this diagonal weighting matrix W and write it instead GT WG and then W times G. So that's weighted linear least squares, where so W is a diagonal matrix where the kth diagonal entry is 1 over the variance of the kth measurement. So here we've decided that the standard deviation of our sorry, there should be a square there, it's the variance. The standard deviation of our kth measurement is some fixed sigma over sk. So that suggests that now what we'd like to put our W as, our WKK as, is sk squared over sigma squared. One problem, of course, is that we don't know sk. Sk is the signal. We only have measurements. However, the AKs that we do have are our best guess at the signals. So what we're going to do is to use the kth signal squared as the kth entry in the diagonal entry in WK. And then, so we could, of course, divide that by sigma squared. I'm actually going to drop the sigma squared term. We don't know what sigma is for a start, but also if you look back up to this weighted linear least squares equation here, you can see that because sigma is just a constant factor, it actually cancels out anyway because we've got W inverse here and W not inverse here. And so the factor of sigma squared cancels out. So we can just ignore that, and we're going to set our WKKs simply to be the square of the kth measurement. So let's just finish up then by, I'll implement that quickly in MATLAB, just because it's the standard way of estimating the diffusion tensor that's used today. So let me time it again. Our usual stuff. Only for voxels with decent measurements. So let's construct this W matrix then. So W then is going to be a diagonal matrix with our measurements squared on the diagonal. Very simple. And now we have to compute the inverse mapping. We can't use the trick that we used before of pre-computing it, because now, of course, W is going to be different for every single voxel. So we have to compute this inverse mapping every time around this, this loop. So there's our inverse mapping. Now we can compute X. And plonk it into the corresponding space map. Just a good measure, I'll do the tick-tock. And you'll see that it takes a bit longer this time because we're computing this more complex matrix and inverting it. Now, did I save? What's that figure? Okay, so this was the mean diffusivity map from the standard least squares. Let's have a look at the one now from the weighted linear least squares and see how much of a difference it makes. Okay, so I've plonked it into the same array, so I think this should give me the, there it is. So you can see it doesn't make a huge amount of difference, but there are noticeable differences there. You can see, for example, that the background is blacker in the weighted linear least squares, and that's because we get fewer sort of noisy, there's less sensitive to noisy pixels that give negative diffusivities. I mean, we could just remove those, of course. But it does that more naturally. And actually, if you do look closely, you can see that there are some differences. And the fact that it's theoretically a more correct thing to do, and it's not particularly more difficult or more time consuming to do, means that this is really the standard way of computing the diffusion tensor using weighted linear least squares. It's not perfect because actually the Gaussian assumption that I said was a reasonable assumption. Where did I say that? Up here somewhere. This assumption here isn't perfect by any means. In fact, there are better noise models for the noise on the MR signal than Gaussian. There are Ricean distributions, there are other ones. But using those makes the fitting much more complicated. We will look at that a bit later, but it becomes much more time consuming. So yes, weighted linear least squares remains really the standard way of estimating the diffusion tensor. Okay, that's the end. I shall stop there and I'll see you in person in future lectures.